Thank you very much, Danny, and thank you to the organizers for inviting us and giving us the opportunity to speak here. And thank you to my previous speaker, of course, Prof. Leo as well. And I will show some slides that shows that Ncid even helped with the success of Ncid that I will also cover partially here. So the topic I want to present is a little bit about evolution of a virus. And we, of course, all know that viruses are constantly mutating. And I want to give a little bit of overview. What are the mechanisms and how can we monitor and track it? What is needed in a global effort to achieve that? And if we look at the SARS CoV2 distribution of variants of concern, as you can see here in turquoise, this is the Delta variant. It came up starting from April and very quickly took over the world, became the most dominant strain to date. You see this big red plot together. This was the Alpha variant. This was before Christmas last year, when the UK reported there was a new variant coming up. And you also see that it had a considerable impact, but it was also very quickly replaced by Delta. So a key point I want to make is that the variants didn't start with Delta or with Alpha. You see all this part here. We've been tracking those changes all along. And with we, I mean the global community that is able to contribute with genomic surveillance to characterize what are changes in the virus. And you can see, for example, there was the original Wuhan wild type in light gray here. And there was another parallel strain in southern China. This was the green one here. And then all the orange colored ones are essentially with the spike mutation, D614G. That was one of the first bigger mutations that were fully fixed in all of the circulating viruses. And then you see the change to Alpha and to Delta. You see Delta compared to Alpha really had a much sharper rise. So a lot of the clinical data that Prof. Leo was also showing about the viral load, etc., that probably plays a role. But it's very difficult to predict this from the onset and just looking at the sequence. So we know the virus is mutating, and it's mutating all the time. And whenever you hear there is a new variant, it doesn't necessarily mean that the change really is relevant. What we know is that there are constant mutations, and you can think of it, it's a very long genome, 30,000 bases. And if you make a little change of it, it doesn't really change how the virus functions. But because these random changes happen in the mistakes in the replication of the virus, it lives like a barcode or like a license plate. So we are able to monitor these changes, and it helps us tracing the virus. We can understand how clusters are related to each other, and also how the virus travels internationally. And this is why the license plate analogy is apt, because you can see from which state or which country it more likely occurred. I will show you later some examples how these lineages are there. At the same time, sometimes these changes do change the properties of the virus. And science and bioinformatics can help a little bit in an understanding how is the mechanism to that. Again, also those changes are tiny, and you can compare that if you look at the car. It's not the change in the license plate, but it's the change in the engine size. So instead of 1.6, it's 2.0 liters. So again, a tiny change on the outside, but you can compare it like that. This is the spike protein on the surface of the virus, and in green is the human host receptor. So if you get the mutation that is near the host receptor, it can suddenly bind better. And with binding better, that means it can possibly infect better, and it's not easy to predict, but the clinical data then can be used to corroborate the change in properties, as well as some in vitro tests. So you have these two changes happening, license plate mutation as well as those phenotypic mutations. And the next thing that you need is how do you track the virus evolution, and how do you detect those mutations? And this is where the Ncid platform comes into play. It's a unique sharing mechanism, allows people to submit the data but still retain the rights to it. This means that they can immediately share a new sequence and other people are allowed to see it, but they are not allowed to patent it or use in their publication unless they collaborate or acknowledge the original submitter. And this is something that has been an important aspect that enabled real-time genomic surveillance. There is no other pathogen that has so many genome sequences as this virus currently. If we think of influenza, we have about 20,000 genomes coming in per year, and here we have more than 20,000 genomes coming in per day at the moment. And this is also what happened in the pandemic, that the sharing of the sequences took a very steep rise during the pandemic, and it was enabled by networks that already existed. And this is the part where I also want to highlight Ncid. This is a photo from a workshop that we held at bioinformatics workshop to help people from the WHO network that was doing flu surveillance use computers to interpret and use genomic surveillance to classify phylogenetically and identify mutations. And this is already the second workshop that we did then in 2019 that were helped by Ncid, before also with Dan Tokseng. And exactly these people that you see here, they are from national influenza centers in different parts of the world, were also the ones that were doing the load of the sequencing across the world. So the GISRIS network that does sequencing around the world, and several of them are here, and when Wenqing, for example, was the lead then, really helped to power the genomic surveillance. So going forward, we also look at the quality that we want. It's not just a database where you dump a sequence, but there is the need to register important metadata information. For epidemiological studies, you need a very good understanding of the sampling strategy used, so they can interpret and you can unbiased bit the data. Some countries are sequencing much more than others, and you need to be able to find a good balance. When you use the Ncid database, you also have fields that allow updating things about the vaccination status, about the clinical state of the patient, if you can and want to share that information. And importantly, of course, there are things like the time, the collection date, and the location, which allows to track where the virus is spreading. And the location, of course, is at best at the district level, and of course not going to the home address, etc. Privacy concern is also a very important one. And the way to handle this incoming flood of genomes requires a team across the globe available 24/7. In the Asia time zone, it has been done by a Singapore team that comes from several colleagues that were joining, also from different ASTA institutes, besides BI, my institute, also GIS that helped. And we are looking 24/7 in the Asia time zone, and then we hand over to the South America time zone, for example, where we have the next team helping to curate the data until it's released, usually within a few hours. So we receive a sequence and it goes out within from minutes to maximally 2 hours. And it also requires a big communication back and forth. So with colleagues, for example, there are about 280 exchanges daily talking about the data, improving the data. Sometimes there is a collection date missing. So the third important item that is one of the benefits of genomic surveillance, there can be multiple bioinformatics tools that allow us to create new insights. And these insights allow to create reports as well as to study and identify what are the potential new variants. And this is where the SARS CoV2 evolution comes into place. So earlier in this conference, colleagues also talked about diagnostic devices. So when the China CDC shared the high-quality genomes on Ncid on January 10th, it was immediately possible for many people to create diagnostic kits and tools. And one could see how the virus could be detected. But looking at the evolution of the virus is also important, because there can be changes that can affect some of the sites, the regions that these PCR primers recognize, for example. And one of the examples that happened was that one of the primers from the China CDC was not recognizing well some of the samples that came from Europe a few months later back to China. And through the genomic surveillance, it was very quickly picked up, and the assays adjusted for this to work again. It's also interesting to note that from the Drosten group, they published nice primers before, including one on the RDRP side, that the Delta variant actually does have a small mutation at the end of one of the primers, which also means it's good to be adjusting that. And most importantly, many of these diagnostic kits are not using a single probe to detect, but they are targeting multiple sites. And these multiple sites allow robustness so that no actual case is being missed. The next thing that we can do is with the real-time surveillance, we compare the strains with each other. We can look at the genomes in a phylogenetic tree. And this tells us a little bit about the transmission dynamics. It tells us where it came and how they relate to each other. And the technology to do that has also improved. There is a very popular way of viewing trees that is also available on the Ncid site. And, but it's limited to 3,000 genomes. So we are also working with other colleagues that allowed to scale up the trees from 6 million to 3 million or 5 million, as we have now 5 million genomes in the whole database. And working with these big trees required a lot of new software developments. So having those trees, apart from seeing the bigger picture, also can help with contact tracing. You can understand a bit where the immediate closest sequence came from. Is there a shared marker? So you can, if you know there is a shared marker among cases, they are more likely to be related, being part of the same transmission cluster. The diversity that you see in the tree also tells you in how many independent clusters are there in your country, for example. And one of the interesting tools is, of course, you want to know what is the closest global neighbor of your genome sequence. And sometimes you also can see where it came from internationally. And the next step is, after knowing more of the context of the nearest neighbors in the genome tree, what are the mutations in the sequence? And this goes back to the example that I showed. Most often and most likely, any mutation will be at the level of being neutral, being more like a license plate mutation. This license plate mutation help positioning it into the tree. But we also want to know what are the mutations that change the engine size of the virus, like the D614G or some of the mutations that alpha had, like the N501Y mutation near the receptor binding site. And this is where bioinformatics tools like the one that we developed here, the COF server, also mentioned by Danny in the introduction, comes into place. So this happens within 1-2 seconds. You can get the result, mapping all the mutations of any new sequence into the 3D structure. And it tells you, is it in a known site that has a phenotypic effect, or is it not well studied before? And what you can see here is some literature reference, how this mutation appeared before, was studied before. So it's very important to integrate this data. It's very important to map this to 3D crystal structures. Besides the host receptor, another very important aspect nowadays with the vaccines available is also, are these changes happening near antibody binding sites? So this is important because if there are many of these mutations in the antibody binding sites coming together, then you have reduced vaccine efficacy as well as more potential vaccine breakthroughs. And having this in place is one thing, it's good to have, but seeing it work is, of course, always a very important aspect too. And this is an example how these tools looked like when the Delta variant came. And the Delta variant from India also came through imports to Singapore at that time, through imported cases without real community transmission early on. But because we could see in the phylogenetic tree that among all the cases from India, there was a sublineage that seemed to be evolving faster. And we saw there were some changes, and these changes happened to be in the receptor binding site, and also in the N-terminal domain, that is one of the antibody recognition sites. So without knowing that it would be, let's say, potentially higher viral load, but we knew it was something that has some of the features that viruses use to become better, to become more successful. And apart from our local colleagues, we also discussed this with Indian colleagues early on. And you can see how sensitive the system is, because we talked to them in April when Delta wasn't, it wasn't even called Delta back then. We told them this strain here was the one that maybe they should look out for. And indeed, it became the Delta variant here. So, of course, then you may want to, it's one example, and you want to know what will be the next Delta. That's a good question, and unfortunately, no one can really know. However, the system that I described, with the global community sharing the data via Ncid and having good analytics tools coupled to it, is one of the ways how to do that. And one way of classifying the viruses is with the Pango lineage and nomenclature system. And this plot here shows everything that is within Delta is here with color, and everything that is non-Delta here. And you can see what are the sublineages within Delta. And the sublineages within Delta mostly and by far are characterized only by these license plate mutations. So you can distinguish them by name, you can assign them different names, but this doesn't mean there is a change in virulence or other change necessarily. This classification does not consider these features. It's purely based on being these license plate mutations. And what is interesting, of course, when you see the distribution, you can also see that the virus again has become different in different parts of the world. So, for example, in Africa, there is AY32 as a dominant and AY45. In Asia, there is AY23. And I will go into a little bit more detail there. And because Singapore has such a strong surveillance system, we also did report more. And when you report more, and there is like a license plate mutation, then you can even get your own lineage, like this AY23.1. This lineage is not unique to Singapore alone. It's in the region. And in the region, if you're the country that sequences the most, then it looks like that it's the most dominant in your place. However, the changes that are there to give the classification of a new lineage, again, are simply this license plate mutation that it is not known that they affect any properties of the virus, really. And similarly, in Australia and New Zealand, they had their own AY39.1 variant. And you also see that there was a change in AY43 in South America and AY44 in North America. In the US, you have in the different states, East Coast, West Coast, also different variants. For example, AY25 is more common in California. And what is important is these changes and these labels of the Pango lineages are important for us to take stock of who is rising and who is coming down. But what is also important is that we continue looking at where are not just the license plate mutations, but where are the engine size mutations. And this tool that we have running inside Ncid takes both into account. It takes into account how fast a certain type of virus is rising based on the combination of relevant mutation, like constellations of mutations, and also if the sites that are changing are actually relevant, for example, being near the host receptor binding site or being in the known antibody binding site. And by adding this information, we can stay up to date and detect what are the new candidates to look out for. And this is indeed part of several groups' input. This includes the Virus Evolution Working Group using a similar approach. This is also in collaboration with CEPI and Public Health England, or HSA, now with the Agility Project. So both for local cases as well as for international cases, when such a new variant occurs, these are immediately being shortlisted for experimental testing, because this assignment of potentially relevant mutations still needs the experimental test and the clinical verification. But the global surveillance system and the real-time sharing with Ncid allows that those variants are coming into the pipeline to be tested very early, so that if they actually happen to rise very sharply, people will already know and have results, how this affects vaccine efficacy, and eventually would require policy changes. And the one that is ranked highest here in the October table is the EY 4.2 that was mentioned. And this is because of two new mutations that additionally were added. And it's interesting if you look in the tools that one of them, the Y145H, complements a cluster of mutations in the N-terminal domain, which is one of the antibody binding regions. There was no change in the receptor and antibody binding region up here, but there was another change here, the A222V mutation, which, if we look historically of where those mutations occurred, also happens to be one that was the marker mutation for an outbreak that happened in Spain last summer, where a lot of travelers brought it back and spread it through Europe. But this was a genomic variant that very quickly died out. So this happened before alpha, beta, gamma, and it didn't even have its VOC name, and no change in properties were really known. But this is just to illustrate how important it is to have real-time surveillance constantly going in all parts of the world. And once they are designated to be a variant of concern or of interest, then we can track them very closely. And doing so with the Ncid sharing platform, I think, has been one of the big successes in this pandemic, and it requires a lot of people to help. Of course, also WHO and the Ncid surveillance network, but also here from the Singapore side, there are important contributions, and these range from Ncid to ASTA, and of course the Singapore lab doing the sequencing here, like NPHL. So thank you very much.
Right, thank you, Sebastian. We have a couple of minutes. I have a couple of questions from the audience. Maybe I'll ask you the simpler one first.
Yeah.
And it asks here, many Singapore ART positive cases are now not PCR tested. So would there be changes in circulating strains, I guess in the proportions and so forth, that is unknown because of that?
Yeah. So if there is community transmission, it is very likely that it's always the same strain. So it is happening only if you get people coming in, traveling in, that is more likely that you get new lineages and variants. So I would think that the proper way, and that is being done here, is to do sentinel surveillance, which is sampling. So the numbers that are being tested and actually sequenced are sufficient to tell if there would be such a change. And we clearly do see that with the data that we have, and for example, the AY23.1, the methods are very sensitive to pick up any relevant changes.
Okay, great, thanks. The second one is by Julian Tang. It's a very technical question. Maybe you can give a short answer and then maybe get offline with Julian after this. Mm-hmm. For the phylogenetic tree construction, do we need to take recombination into account? Initially we did not because recombination seemed rare, but what are you seeing now, 2 years later? And also, for your GSID default tree construction, which model of evolution are you using? Is it just the HKY85 or GTR? So maybe a brief one to that and follow up with Julian.
Yeah. So thanks, Julian. And I think recombination is possible for this virus, but it's happening very rarely. So there were some papers that are still being under review that made a case that they found some cross lineage recombination. I would say it's not one of the bigger evolutionary mechanisms in this pandemic. And for the second question, to make these very big trees, one needs to use very fast methods. It's a maximum likelihood method based on IQ tree, and that uses a GTR model.
Excellent. Sebastian, thank you very much for your excellent talk.
Thank you.